My name is Bonnie Ramsey. I'm a professor of pediatrics at the University of Washington School of Medicine, and I'm talking today about therapeutics development using the cystic fibrosis community as a model of success. So I'm delighted to be here, and as Fiona said, I am a pulmonologist, a pediatric pulmonologist, and for the last over 30 years I have been taking care of patients and families with cystic fibrosis. And I've had the honor of being not only part of their lives, but also part of two major drug development processes. Now, I never intended to be an entrepreneur, and actually when Fiona asked me, I said, but I'm not an entrepreneur. I was interested and passionate about trying to change the lives of the patients by getting new drugs. And so I think what I'd like to have you take away from this is, even though it's a huge process, you can be a key part of it, and you can enjoy every minute of it along the developmental pipeline, which I'll show you. I have to disclose any grants and contracts that I have. I am the director of the Therapeutics Development Network, which designs studies for cystic fibrosis around the world. And as such, we help a lot of companies, and all the contracts are through Seattle Children's Research Institute. I don't take any of the funds. Personally, the two that are highlighted, Novartis makes Toby and Vertex makes Kalydeco. I will be discussing them. Just to make it clear to you, I was one of the co-inventors on the patent of Toby, which is Tobramycin for inhalation. I initially received $10,000 because they had to give me something, and in 1997, but had no stock. And for Kalydeco, I received no stock or reimbursement. So what does it take to develop an FDA-approved drug? I will come back to this, but if you start at the level of high throughput screening, which you see here, where you have many different chemical entities, and you're trying to look for what they call a hit, which means you have some laboratory assay. In this situation, it happens to be chloride movement, so an ion channel. And if you have movement, then you get a fluorescence, and that's a hit. So to move from that to actually having a drug, what is going to have to happen? Well, there's a lot of things to consider. Number one, not every chemical hit that you're going to get in a lab tray is going to be a drug. So can it be modified so that it's druggable? And that's what medicinal chemists do, working with a big team. So that's the number one question. Number two, can it even be manufactured? I mean, many people have wonderful ideas, but there's no way you're ever going to be able to make it at mass production at a price that anyone could pay. Now, many of you are thinking, do you know the price of Kalydeco? Yes, I do know the price of Kalydeco. Is there a market for the drug? It doesn't do you any good if you can't define who your market is and they can't sell it to someone. And how are you going to give it? I'm going to show you one drug that is inhaled, and the other drug is a pill. But you have to decide which way is going to be best for your target. Does it have an acceptable profile? And by a PK profile, we mean, can it be absorbed? Where does it go? How is it metabolized? And how do you get rid of it? So the FDA wants to know everything that ever happens to that drug once you give it. What's your target organ? Do you want it to just go to the brain? Do you want it just to go to the lung? Or is it systemic? Is it safe and tolerated? Is it benefiting the patient? Now, this is an important concept. I'll come back to it. There's a difference between a biologic effect and a clinical effect. You can have a biologic effect, but if it doesn't help the patient, it's not meaningful to the FDA. And is there an appropriate patient population? I think I mentioned that. So you can find lots of variations on this if you go on Bing or Google. And this one, I thought it was a nice one. I actually got this off of University of Wisconsin's website. But it shows you the phases of drug development. So at the far left, in the discovery phase, you'll often have thousands. For Kalydeco, I think they screen something like 100,000 compounds. And then you have to go through many iterations till you find something that's druggable. By that time, you're down to 100 to 200. And then you have to go through testing in animals and in different in vitro cell models to show whether you can get it to a dose that's reasonable and it's safe, it doesn't, kill animals or whatever. And so that's the preclinical phase. And at that point, you start thinking about human trials. And if you look at the bottom, you'll see the hatched area. So it starts with phase 1, which are tiny trials, usually maybe 10 to 20 patients, to just see whether you can get the drug absorbed and is it safe. And then you go to the second phase, which is really, to me, the most exciting studies you do, because this is where the rubber hits the road. This is where you show whether you get a biologic effect and whether you have a potential candidate. At the end of phase 2 is what we usually say is the moment of proof of concept, and I'll come back to that. And then you go to phase 3, where you get the huge trials, which could be 1,000, maybe 10,000 patients. Depends on the patient population, and those are very expensive. This whole process is, if you're lucky, 10 to 15 years. And if you're lucky, it's only $500 million. Sometimes it's going to be up to $750 to $800 million. It's a very expensive proposition. But most of the money is going into the later phases. Proof of concept, it occurs, as I said, between phase 1 to 2 and phase 3. It's a very strong signal that you've got a winner. And why is this important? Because this is when a company decides whether they are going to invest in the large-scale manufacturing and the large-scale clinical trials. They've got to put at least $100 million on the line, and so that's a big decision. And what they go by is it safe and tolerable? Does it have good pharmacokinetics? Did it get into the body at levels that you could actually manufacture? And was it excreted, and so forth? You have to show a biologic effect, at least, and I'll come back to this. Ideally, you want to show how it's helping the patient. You don't have to have that, but it sure helps if you've got something along those lines. So I'm just going to take maybe 5 minutes to tell you about cystic fibrosis, and then I'm going to present 2 case studies of drug development that I have been involved in. Cystic fibrosis is a genetic disorder. The prevalence in the United States is 30,000 individuals, and there are 70,000 identified worldwide. Because of large national and international registries, we actually, this is a population that's very well tracked and understood. It meets the definition of a rare disease. Now, a rare disease is any illness that has less than 200,000 individuals in the U.S. Different countries have different definitions, but it's fairly close to that. And that's important because you get there are special congressional acts related to rare orphan diseases, and you get special help with the FDA, you get prolonged exclusivity of your drug. So there are advantages to being a rare disease. Unfortunately, by definition, there's fewer patients for doing studies, and there's fewer patients for a market. So those are the downsides. The life expectancy is about 38 years now. That's a dramatic increase from when I started 30 years ago. It's actually an increase of about over 20 years since then. The median age is 26 years. It is lung disease, which is the primary cause of morbidity and mortality in this illness today. They also have pancreatic and liver issues, but the mortality is from the lung disease. Cystic fibrosis is caused by a gene from which a protein, which is called cystic fibrosis transmembrane regulated protein, CFTR, is coded. And CFTR is an ion channel. It sits in the lining cells of the lungs, along the airways, in the lining cells of the pancreas, the liver, and several other organs, but those are the key organs. And what is critical is that in the airway lining cells are called epithelial cells, and there is a layer of salt water that sits on the top of them, and then on top of the salt water is a mucus layer. And constantly there are cilia that are clearing the lungs. So we breathe in all sorts of dust and particles every minute of the day. And this mucociliary clearance keeps our lungs clear. You're not even aware of it unless you have a cold, and then you become aware of it because you have mucus. But that layer of salt is very, very closely monitored. It stays within a micron of where it should be at all times. And the pump that is very responsible for that is CFTR. If you don't have CFTR, that salt level starts going away, which is shown in that lower airway picture. So all of a sudden you don't have this layer of salt water. It becomes dried out. It's like a bathtub where the water went out, and it becomes dried out, and the mucus becomes very thick. And then you have trouble coughing it up. And the problem with mucus is that mucus is something that bacteria love to grow in. And if it becomes thick and obstructed and you can't get it out, then bacteria get in there and they live in there and they set up infections. And you get infections that you cannot clear. And they're very specific bacteria. One of them is Pseudomonas aeruginosa. And the airway will simply not clear it. And those infections lead to lung damage over many, many years, which is irreversible, and that's the reason that patients die of cystic fibrosis. So that if at the bottom you see a normal airway is that open white, that's a clear airway you could see through. On the left is CF, and it's a plug. It's a plug of mucus with bacteria in it, and that is the basis of CF. So I'm going to talk about two case studies. The first is at the bottom, and that is at the level of treating the infections in CF with an inhaled antibiotic. In this situation, this was an observation made here at the University of Washington and Seattle Children's by Dr. Arnold Smith and myself. And it was looking at inhaled antibiotics. And so that's the intellectual property was here, the initial development to phase 2 was here, then a company came in, and then it went to approval. The second case is dealing with a treatment for the salt channel, so making the abnormal CFTR work properly. In this setting, the company had the intellectual property, but working with the CF Foundation and the CF community, we worked as a team to get approval of that drug. The Toby study started actually in the 1980s with Arnie Smith making a seminal, very simple observation. He took sputum from a patient with CF, went to the laboratory, and said, how much aminoglycoside, which is Tobramycin is a kind of aminoglycoside, does it take to kill Pseudomonas in CF sputum? And he found that if you don't add any antibiotic, sputum is a place bugs love to grow, so that's the green line. They will just take off in it. But you had to get up to 25 times the dose that you would normally think you had to use to kill bacteria in CF sputum. And so the problem was CF sputum was binding up all the bacteria, so you were just dumping it in there. So what we were doing, treating intravenously, was basically not even getting close to what the patients needed. So we had to come up with a way that we could increase the dose 25 times and not be toxic to the patient. So the only way to do that would be to have them inhale it. Now the good news is Tobramycin is not absorbed through the lungs, so you could put a lot in there. So we had to come up with a proof of concept. Proof of concept was that you had to get high concentrations into the lung. We had to show that. It had to be safe. You had to be able to tolerate it. It had to kill the bugs and yet make the patient better so they could breathe more easily. This picture shows you our first attempt. Now this is a classic example of academics doing drug development. And so there weren't the kind of inhaler devices or nebulizers they have today. So we devised this thing where we had this water bath and we had all this tubing, and then the reason the nose plugs is because there was so much flow that it would come back out the nose unless we had a plug on. So we developed this and it worked. It got very high concentrations in the lung. We killed 99.9% of the bacteria. You say, why not 100%? But actually, that was much better than we had done previously. And it showed a marked improvement in lung function. And they were striking enough that it was published in the New England Journal. Problem was, it didn't meet any regulatory requirements, and so no insurance companies would pay for it. And we didn't have, at that point, we didn't have any IP protection or a patent. It wasn't clear who owned the clinical data, and we hadn't done the animal tox data because this was a standard drug that was available for IV use. So we had created this totally impractical system that wasted 90% of the drug because it was all blowing out, and it was not portable. So from our standpoint, we thought we'd done something great for the community. We were terribly excited about it, and then we realized we couldn't get it to the patient, so it was really of no value. That was very disheartening. So the CF Foundation went out and found us a partner, and it was actually, there's the irony, there in Bethesda, the partner they found for us was here in Seattle. And it was a small company called Pathogenesis. And they were willing to look at it because they saw the data in the New England Journal, and they said, this is phenomenal proof of concept data. The person there, a person named Bruce Montgomery, actually had some experience in developing inhaled antibiotics. And they did a business plan, and there was an acceptable market for it. They felt that this could be viable, and so they could get venture capital to buy into it. So with that, we started a registration process with the FDA. Pathogenesis took on getting a new formulation in a much smaller machine. We did all the design of the studies, to the phase 2, phase 3. We decided on the outcome measures. We did all the analysis of the data. Pathogenesis did the manufacturing stability, and they did all the regulatory communication with the FDA. And then CF was the matchmaker and did give the initial support. So in the end, we did a large phase 3 study. Actually, it was the biggest one that had been done in the CF population at that time. It was also in the New England Journal about 6 years after the first one. And the outcome measure that the FDA felt was approvable for benefit is that we decrease the number of hospitalization by increasing the time to need for IV antibiotics. So with that, we were able to get FDA approval in 1997. And the little machine you see there is exactly what still is used today. And it's given for 28 days, and then there's a 28-day off period. That was set up to try to reduce the emergence of resistance, to give people a break. And it was indicated for CF, although it certainly has been used in other chronic lung disease since then. Just let you know that last week, this Pathogenesis was eventually bought out by Novartis. Novartis now owns it. Last week, Novartis got approval to now switch this to a dry powder inhaler. So it took them 16 years to get the next formulation, but I'm actually delighted. Why was Toby so important? Well, it provided a proof of concept that inhaled antibiotics could work. You have to understand there had never been a formal approval of an inhaled antibiotic in any illness before this. And so for all other inhaled antibiotics, they used the Toby pathway, both here and also in Europe. In fact, Europe requires that you test it against Toby, any new drug. It did improve the lives of patients with cystic fibrosis. We weren't able to look at mortality, but there subsequently have been studies using the National Registry, and the chronic use of Toby did have an impact on survival. It showed, most importantly to industry, that a small market could be financially successful. There has been hundreds of millions raised, of sales over the 16 years. It introduced the CF Foundation to the concept of effective partnerships. It totally changed the approach of the foundation, which I think is changing the approach of many other nonprofits now. And that was to develop the idea of venture philanthropy. And so the year after Toby was approved, so the foundation was part of the patent, and they had some funds from the Toby patent, and they turned it in 1998 into the Therapeutics Development Program. And the whole concept of this was to de-risk drug development in a rare disease. Because we knew that if you just go to industry, they're going to say you're too small of a market, we're not interested. But if you can bring a package that reduces the risk by first funding the discovery phase, by helping with the high throughput screening, help with bringing the people of expertise in, and then as you go into preclinical, have people help with preclinical. One of the people who has worked with the foundation for years is Danny Liggett, who is the head of comparative medicine and has given consultation to many investigators on preclinical, particularly in inhaled therapies. And also developed this TDN, which is a network of 77 sites around the country, and now there's a sister network in Europe and another sister network in Australia, where the trials are all conducted, where there are investigators that understand the outcome measures, etc. From this came the second case study. Now, I had the honor of basically leading the Therapeutics Development Network, which I have for 16 years. And one of the investments, the biggest investments, has been with, originally it was a company called Aurora, which is why you see Aurora written up there, which was a small startup in San Diego that did high throughput screening for ion channels. Now, most high throughput screening wouldn't look at ion channels because it's pretty hard to do high throughput. You can do low throughput, but not high throughput. And so they specialized in it. It was, so then Vertex bought out Aurora. And so the foundation funded the original screening, and they put together this development committee which included academics, the CF Foundation, and then people from Aurora Vertex to start thinking about a whole development program. And I'll show you that in the next slide. They screened for about probably 8 years. And when it was started, they thought, well, we'll find one drug that'll be the cure for CF. It will be able to repair the CF protein. So the CF protein is made, in the endoplasmic reticulum, then it's got to be trafficked up to the surface. It has to sit in the surface of the cell, and then it's got to open up and be a chloride channel. And when they started screening, they thought they'd find one drug, but it became clear over time that there really were going to be two classes of drugs that were going to be necessary. And these two classes were potentiators and correctors. So potentiators are drugs that open channels, chloride channels. And so in this picture, you can see the channels are those little yellow spiky things. And then if they open up, then you get chloride movement. The correctors are drugs that traffic, they're often called chaperones. They traffic proteins up to the surface. And so if you have the most common mutation in CF, which is delta F508, the protein isn't even at the surface, and so you have to have something to get it to the surface. Now, naturally occurring, there is a type of mutation in CF called G551D, where the protein is able to get to the surface, but it can't open. Whereas in the majority, it doesn't get to the surface and it doesn't open. It doesn't do either of those steps. The drug development program that was set up was a combination between the TDN, which was investigators from around the country, the CF Foundation and Vertex, and let me just go over which each one did. And then this joint development committee was probably one of the most exciting things that I've ever been able to be a part of, because they actually, even though I'm a clinician, they actually brought me in at the point that they were doing the screening, the high throughput screening. And when I first got there, and this went on for years, it was highly confidential, I thought, why would you have a clinician here? But it was so powerful to have the entire team there the whole time, because it kept everybody thinking about what was the end goal. So the academic group, we were to define the patient population, identify the biomarkers and clinical endpoints, design the studies, conduct the studies, and do the data analysis. The foundation did de-risking in terms of initial funding. They formed and ran this development committee, and they went out and found experts. You know, every time we'd go down there, you know, we need another cell type, and they'd run out and find somebody who had a cell type. You know, it was really pretty impressive. They also supported the TDN. And then Vertex did all the preclinical development, so the drug screening, the formulation, the animal tox, the manufacturing, regulatory, similar to what Pathogenesis did. They funded the clinical trials as well. So the proof of concept, let me just mention, the potentiators happened to be a little bit ahead of the correctors. So as these different screens were coming out, the potentiators became available about 2 years before the correctors. So there was a huge decision to be made. Do we wait till the corrector comes and develop both of them together, or do we try to develop the potentiator by itself? Now, in drug development, it's hard to develop one drug. To try to develop two together is insanity. And so there's a real advantage to getting one approved first. The problem was you had to have patients that had the CFTR up at the surface. And fortunately, through all the networks, we were able to identify about 2,000 patients in the world, not in the United States, in the world, who had this mutation. So it was felt that they could take the risk to go into this tiny, tiny population. So we started with the potentiator, which was called VX770. Vertex's role, they had to show that, you know, they had to do the manufacturing, but they had to show that it reached the blood and then the tissue where it was going to be effective in the lung, and they had to show it was safe. Now, the TDN, the clinician's job, was to say, how are we going to know that there was a biologic effect? It's different than tissue culture. So there were two measures that we felt you could use. There's the sweat test. So patients with CF have salty sweat. That's the old diagnostic test. So that's obviously related to salt movement. And so the sweat test. The other is you can measure a potential difference or, you know, voltage in the nose, which is related to chloride and sodium movement as well. So we thought we would choose those two biologic measures. We chose the G551, it's not two, it's supposed to be one mutation, as the target group. And then ideally we thought, well, we'll just look at lung function at the same time. So this is what we banked on. So we did a proof of concept study in, I think it was about 70 patients total. It was a dose escalation. And so sweat test was going to be our proof of concept, and this was in the New England Journal. And let me tell you, to have a small phase 2 in New England Journal, you better have strong results. So the sweat test was, I mean, it just knocked our socks off. So the sweat chloride in CF is greater than 60, and most patients with CF, it's up around 90 or 100. On this drug, the patients with G551D dropped their sweat chlorides to around 30 to 40, which is in the normal range, in a matter of about 3 weeks. So we had proof of concept. We then went on to a big study, a year-long study, and there we wanted to look at improvement in lung function and decrease in pulmonary exacerbations. And so this was in the New England Journal in 2011, and it had a 10 to 12% improvement in lung function, and these are in totally healthy people. So it's not like they're sick and we made them well. These were people who were already stable, and we improved what their status was. You can see that the improvement's very, very rapid. By 2 weeks, it's already happened, and then it's absolutely stable. There's now open label data that goes out. This goes out for 6 months. There's data that goes out for an additional 2 years, and it's still there. It's still stable. For some reason, it doesn't take another blip, but it remains very stable. I'm looking at Pradeep, who's sitting here, because he's just been in Ireland, where they just initiated this drug, and he's seen the remarkable change that occurs when this drug is started. So that change in lung function, and then also it decreased the number of pulmonary exacerbations or pulmonary infections as well. We don't know its impact to date on Pseudomonas or other bacteria, because that was not looked at in this study. With that information, the FDA approved the drug in January. They actually approved it 6 weeks ahead of time. And so it's now available worldwide, and I think the last time I looked, something like 70% of all patients with this have gotten access to it, so it's been pretty rapid. Now, have to acknowledge the fact that this is only 4% of all individuals with cystic fibrosis. So that means 96% of people with cystic fibrosis don't yet have access to this type of drug. And that doesn't make anybody happy, including the CF Foundation, myself, or Vertex. And so there is a huge process going on, also through the Therapeutics Development Network, to try to make sure there's something for the remaining 96%. So the first thing that's happening is right now they're testing Kalydeco alone in other mutations that are very similar to G551D, such as R117. And so those studies are going on. That's not a huge number, but that will increase the number of patients from about 4 to 8%. The bigger project that's going on right now is there is a corrector that is moving through clinical trials, and that's called VX-809. And it is now starting as of, I think, in 2 weeks, phase 3 worldwide studies. And so in that setting, they actually have created a combination pill with the Kalydeco and VX-809. I'm one of the lead investigators in that study, and it will be enrolling 1,000 patients worldwide. And the initial results are encouraging. I don't know that it will be the same as G551D. It's very interesting. There's less of an impact on sweat test and more, but seems to have an impact on lung function. So it's not clear why that is. And so that will cover 65 to 70% of the patients. And now the foundation has just started a whole new initiative working with multiple companies on coming up with different solutions for the additional 30% of the mutations. So it's a huge step, but we're a long way from getting there. So I just like to end, and then we'll open it up for questions. But I can see when you see that initial map going from 10,000 chemicals down to one product, $500 million later, it's like, well, how could I possibly do that? I think that, you know, when I started out, I had no idea that I would be in drug development. I think I got into it, as I said, because of my passion for trying to do something for the patients. And just the process itself was enjoyable. I will tell you, I've worked with many, many companies, and I've been involved in many, many failures. So you're seeing the two, there's like 80 on the other side. So you have to understand there's a balance here. But nobody can do it alone. Part of the fun is you work in these huge teams. Again, when I was able to be part of the Aurora team, working with people I would never have met in my life and getting their input was just so rewarding. But you need to ask for help early. You could see with Toby, when we got started, we went down the wrong road. We happened to come out okay in the end, but there was a lot of wasted effort there. But back then, they didn't have what we have now. So now there really are people to reach out for. Obviously, the Center for Commercialization, if you have an idea, they will get you plugged in to people that can help you. The Institute for Translational Health Sciences has what's called its Drug Discovery Advisory Committee, which will take your idea and connect you. Often, for example, they may connect you with a fellow in the School of Business who will put together a business plan for you. We've had fellows from the law school to help with contractual issues. School of Pharmacy, one of the people we often work with is Rodney Ho, who will be very helpful in formulation. So you just need to step out and ask for help, and it's there for you. So with that, I'd just like to thank you for your attention.